Okay, let's unpack this. We're diving into what is, I think, one of the most exciting frontiers in biology right now.
But also computationally, one of the most, well, brutal ones maybe.
Exactly. Single-cell multimodal omics. We're not just looking at, say, gene expression anymore.
Well, we're profiling multiple layers simultaneously. Think RNA, proteins, even the epigenetics.
All within one tiny single cell. It's kind of mind-blowing.
It really is a revolutionary shift, capturing all these different data streams from the same cell. It gives us just unprecedented power.
Power to do what, exactly? Like, what questions can we answer now?
Well, fundamental things. Defining what a cell actually is, its identity. Tracking how cells make decisions, their fate. And crucially, dissecting disease mechanisms at a resolution we just couldn't get before.
But you hit the nail on the head earlier with computationally brutal, taking all that diverse data
Yeah, these massive streams.
and making sense of it together. That seems like the real bottleneck, doesn't it? The integration part.
That is absolutely the crux of it. Single-cell data integration. It's a huge challenge.
So before we get lost in the algorithms, let's clarify what we're actually trying to combine. What are the main modalities, the main data types?
Good idea. We're primarily focused on three really prevalent types right now. First, there's gene expression, so the RNA. That's like the cell's instruction manual being read out. Things like CIT-seq measure this.
Okay, RNA. Got it.
Second, surface protein abundance, often called ADT. This uses antibody tags to give us a direct count of key proteins right there on the cell surface. Real-time functional info.
ADT proteins on the surface, right.
And third, chromatin accessibility, or ATAC. That's the epigenetic layer. It tells us which bits of the DNA are physically open, accessible for genes to be switched on.
So RNA, surface proteins, and DNA accessibility. Wow.
And technologies like ShareSec or even the trimodal T-seq are generating this stuff.
It sounds like trying to build a single coherent map from, I don't know, satellite photos, street view images, and maybe infrared scans taken at different times. You need a system to stitch it all together.
Precisely. And that's why we need a guide. Our deep dive today is really built around a major study that did exactly that heavy lifting for everyone.
Oh, okay. What did they do?
They took 40 different computational integration methods and benchmarked them systematically. They used 64 real data sets plus another 22 simulated ones, so a huge amount of data.
Okay.
And they tested these methods across 7 common analytical tasks we actually do with this data.
So this study basically provides the guidelines we've all been needing to figure out which tool is actually the right one for my specific data.
That's the goal, to cut through that complexity.
All right, so where do we start? The study apparently classified the problem based on the data structure itself into 4 categories.
Exactly. And understanding these categories is really key, because the best method absolutely depends on what your input data looks like. It defines the challenge.
Okay, lay them on us. What's the first category?
The first, and probably the simplest, is vertical integration. Think of this as one single data set, maybe from one experiment run.
Yeah.
And within that data set, multiple modalities are perfectly paired for each cell. Like you measured both RNA and ADT from the very same cells in the same batch.
Okay, so everything lines up vertically for each cell. All the info is there, just needs smart combining.
Precisely. It's complete, just needs integration.
Makes sense. What's next?
Next up is diagonal integration. And here the complexity jumps up immediately.
Okay.
Now you have multiple data sets, maybe from different experiments or labs, but crucially, they have non-overlapping modalities.
Non-overlapping, like what?
Like trying to align an RNA-only data set from Lab A with, say, an ATAC-only data set from Lab B. They measure different things in different cells.
Wait a minute. If they don't share features and they're not from the same cells, isn't that like comparing apples and, well, oranges? How can you possibly align them computationally?
That's where the magic, or maybe the heavy lifting of the algorithms comes in.
Mm-hmm.
They don't align features directly. Instead, they try to find underlying biological signals that might be common, like shared cell types. So the algorithm essentially says, okay, this cluster of cells in the RNA data looks like a macrophage based on its genes, and this cluster in the ATAC data also looks like a macrophage based on its open chromatin. So we'll assume they represent the same biological reality and project them into a shared space.
So it relies on inferring shared biology. That feels like a pretty significant computational leap.
It is. Definitely a bigger challenge than vertical.
Okay, what's the third category?
It's mosaic integration. This one's interesting. It also involves multiple data sets, but here they have overlapping multimodalities. And the key is using one data set, one batch, as a kind of bridge.
A bridge. How does that work?
Imagine you have a big RNA-only data set and a separate big ATAC-only data set. You can't directly compare them using diagonal integration maybe, but then maybe you run a third, perhaps smaller, experiment where you do manage to measure both RNA and ATAC together paired in the same cells.
Ah, so that paired RNA plus ATAC batch acts like an anchor.
Exactly. It becomes the bridge modality. It links the RNA world to the ATAC world, allowing the algorithm to align all three data sets.
That sounds incredibly powerful. But I have to wonder, if I'm just using public data, am I likely to find that perfect bridge dataset just lying around? Or is this mostly for people designing specific bridging experiments?
That's a really practical point. It can be a challenge. Finding that perfect bridge isn't always guaranteed.
Oh.
And the fact that there aren't as many robust mosaic methods kind of reflects that difficulty. It often depends on having that specific bridge available.
Right. Okay, so what's the final category?
The last one is cross-integration. Here you again have multiple data sets like multiple experimental batches. But in this case, all the modalities are fully matched within each batch.
So every experiment measured, say, both RNA and ADT.
Correct. Maybe you ran 3 separate experiments, and each one generated paired RNA and ADT data. The data types aren't missing within a batch.
So the challenge here isn't missing data, it's what batch effects.
Precisely. It's purely about removing that technical variability, the noise that crops up because you ran the experiments on different days, or used slightly different reagents, or maybe they were done in different labs. The notorious batch effects.
Okay, so we've got the 4 structures: vertical, diagonal, mosaic, and cross. That clarifies the input data. But what are we trying to do with the integrated data? What are the goals?
Right. Integration isn't the end goal itself. The study defines 7 key analytical tasks that these methods are ultimately trying to enable or improve.
Let's list them out.
Okay, methods need to be able to: 1, perform dimension reduction. Basically create those clean, low-dimensional maps or embeddings of the cells.
Right, the UMAPs and TSNEs we always see.
2, achieve batch correction. Get rid of that technical noise we just talked about. 3, support accurate clustering. Find the distinct groups or populations of cells.
Identifying cell types.
4, enable classification. So, assigning identities to new unknown cells based on the integrated map. 5, facilitate feature selection. This is about finding the key genes or proteins, the molecular markers that define those cell types.
Finding biomarkers.
6, handle imputation. Sometimes you need to fill in missing data points, either a whole modality or specific features. And finally, 7, support spatial registration. This is for spatial omics data, aligning different physical slices of tissue into one cohesive structure.
Wow, okay. That's a comprehensive list of tasks.
Wow.
Now we can actually see where different methods might shine or, well, struggle.
Exactly. So let's dive into the benchmark results, maybe starting with those foundational tasks.
Okay, let's start with vertical integration. Remember, this is the easy one. Data is perfectly paired within a single data set. For basic things like dimension reduction and clustering, which methods did well, which ones were best at keeping the true biology clear.
Yeah, for this scenario, whether it was just 2 modalities like RNA plus ADT, or even the complex trimodal RNA plus ADT plus ATAC combination, methods like Seurat-WNN weighted nearest neighbor analysis and also multigrade consistently performed really well.
What made them stand out?
They seem to be very good at intelligently weighting the information from the different modalities. They combine it so that the distinct cell types are preserved and really well separated in that final low-dimensional space, that embedding.
So if your main goal is discovering new cell types, those sound like good choices.
Definitely. They prioritize preserving that biological variation.
Okay, what about feature selection in this vertical setup? Finding those molecular markers. You mentioned a trade-off there.
Yes, this was a really interesting finding, highlighting a key choice for the user. It boils down to specificity versus reproducibility.
How so?
Well, some methods, like Matilda and Scamomat, they're designed to find markers that are specific to individual cell types. Like finding a protein that's only high on one particular subtype of T cell.
That sounds biologically very useful. High specificity.
It is. But then you have methods like MOFA plus UP. It takes a different approach. It selects a single set of features or markers that are informative across all cell types. They call it cell type invariant.
Less specific to one type, then.
Less specific, yes. But the study found that MOFA plus produced feature sets that were significantly more reproducible when they tested it on different data sets.
Ah, so more stable, more reliable, maybe.
Exactly. More stable even if the experimental conditions varied a bit. So it presents a clear trade-off for you, the researcher. Are you prioritizing finding those highly specific maybe rare markers? Or do you need a set of markers that you know are robust and reproducible? Precision or reliability?
That's a critical decision. Okay, let's shift gears to diagonal integration. This is tougher, aligning, say, an RNA batch with a separate ATAC batch. You said this is where a major trade-off really comes into play.
Oh yeah, this trade-off is probably the biggest headache, honestly, in all of data integration.
Why is it such a problem here?
Because you're trying to force these two fundamentally separate data sets to line up. If you push too hard on correcting the batch effect between them,
You might lose something else.
you risk over-smoothing the data. Think of it like using a really strong noise reduction filter on photograph.
Okay.
You definitely get rid of the noise, the technical differences, the batch effect. But you might also blur away the fine details, the subtle but real biological differences between closely related cell types.
So you lose biological resolution to gain alignment.
That's the core tension.
So which methods were better at which side of that coin? Which ones were great at alignment, even if they blurred things a bit?
Yep.
And which fought harder to keep the biological details sharp?
Okay, so for users prioritizing the biological signal keeping cell types distinct, measured by things like dimension reduction and clustering metrics like CLSI, methods like ski bridge and ski joint really excelled there. They kept those cell type differences crisp.
Okay, ski bridge and ski joint for biology first.
Right. But conversely, if your absolute top priority is removing every last bit of technical variation between the batches getting the best possible batch correction score, measured by metrics like KBET or ILSI, then methods like GLUE, Uniport, and Portal delivered stronger results on that front.
GLUE, Uniport, Portal for batch correction first.
Exactly. So the question for you, the listener, becomes, how much potential blurring of cell types are you willing to accept to make sure your different batches are perfectly aligned? It's a judgment call.
All right, let's move into the even more complex stuff. Multiple batches, maybe missing data. Let's tackle mosaic integration first. That's the one using the bridge modality, right?
Correct. Using that shared modality, like a paired RNA plus ATAC batch, to link RNA-only and ATAC-only datasets.
Were there many methods for this? You mentioned it was tricky.
Yeah, fewer methods tackle this, likely because of that need for a suitable bridge dataset. But among those that do, StabMap actually showed really strong performance across the board for dimension reduction, clustering, classification across different kinds of data combinations. A good all-rounder, it seems.
StabMap. Okay. Any others?
Yes, if your specific setup involved RNA, ATAC, and that RNA plus ATAC bridge, then a couple of deep learning methods, specifically multi-sub-edge and Cobalt, were consistently strong performers.
Multi-6 and Cobalt for that specific RNA-ESPA bridge scenario.
Got it. Now, what about imputation in these mosaic datasets, filling in the missing data? There's a surprising finding there, something about how good the imputed data looked versus how useful it was.
Yes, that was quite counterintuitive. So some methods, maybe like total, might generate imputed data, let's say, predicting missing ADT values that look structurally very similar to the real thing if you had it. It scores really well on math-based metrics like structural similarity, SMSE.
Which sounds great, right? You'd think so. But other methods, for instance, SciPen, might produce imputed data that scores lower on those structural similarity metrics.
Okay.
But paradoxically, that SciPen imputed data sometimes led to better results in the downstream tasks that actually matter, like clustering the cells correctly or classifying them accurately.
So making the imputed data look mathematically correct doesn't guarantee it actually helps the biology.
Exactly. It's a crucial lesson. The imputed data needs to be biologically meaningful, not just structurally similar. It has to feel right for the downstream analysis, not just look right on a formula.
Fascinating. Okay, let's move to cross-integration. This is where we have multiple batches, but all the modalities are present in each batch. The main challenge is just batch effects.
Right, like 3 experiments, each measuring RNA and ADT.
So for that common RNA plus ADT scenario across batches, what worked well?
Again, Total UI showed up as a strong performer for that bimodal case.
Okay, Total UI seems versatile.
It does. But if you scale up the complexity, say you have that trimodal data, RNA plus ADT plus ATAC, and you measured it across several different experimental batches.
The ultimate challenge, maybe?
Pretty much. In that really complex scenario, multigrade proved to be the most robust option overall, across various tasks.
Multigrade again. It seems to handle complexity well.
It does seem quite robust. But we still see that batch correction versus biology trade-off even here. For instance, SCMDC was noted as excelling in dimension reduction, preserving biology, while SCOMAT provided stronger batch correction in this cross-integration setting.
Still no single winner, always a choice.
Always a choice.
Okay, final task. Spatial registration, aligning those spatial transcriptomic slices. Treating location as another modality, essentially.
Yeah, really hot area. We're trying to create a common 3D coordinate framework from multiple 2D slices of tissue data.
What did the benchmark show here?
Interestingly, methods that rely on simpler linear transformations perform very consistently well, specifically PACE-C, particularly with center alignment, and PACE-C2.
Linear methods, not fancy deep learning.
For this task, it seems so. It suggests that for aligning sequential tissue slices, maybe from the same tissue block, preserving the internal spatial relationships within each slice using these linear approaches is highly effective.
That's interesting. Less is more, perhaps.
Perhaps in this context. However, there was an important caveat about the evaluation metrics themselves.
Oh.
One common metric, the spatial coherence score, or SCS, sometimes produced identical scores for all the linear methods.
So, it couldn't tell them apart.
Exactly. It made it impossible to distinguish which linear method was truly best based on that metric. It just highlights that we also need continuous improvement in how we evaluate these methods, especially for newer areas like spatial integration.
Okay, this is all great, but let's talk practicality. For researchers, with deadlines and limited server time, how fast are these methods? How much memory do they chew up? Scalability matters.
Hugely important point. The benchmark did look at this practical overhead, and there were some clear winners in terms of efficiency.
Who are the speed demons?
Well, for vertical integration, a method using matrix factorization called UINMF was noted as being exceptionally fast and needing very little memory.
UINMF for vertical efficiency. Good to know.
And for the more complex mosaic and cross-integration tasks, StabMap stood out again. It generally offered great performance without demanding crippling amounts of computational resources.
So StabMap is not only good, but also relatively efficient for complex cases.
That seems to be the case, yes.
But you mentioned earlier something like 26 out of the 40 methods tested were deep learning based. Were those the resource hogs?
Generally, yes. Many of them often require GPU acceleration to run in a reasonable time.
Right.
And they can consume substantial memory and just take a long time to run. The paper mentioned methods like SCMM and, interestingly, MOFA+, which we noted for its reproducibility as being quite computationally intensive.
MOFA+ is intensive, huh?
Yeah, which could pose real scalability challenges if you're trying to analyze data sets with, tens of millions of cells, which is becoming more common.
Definitely a practical constraint. What about robustness? How stable are these methods if the input data isn't perfect? Like if you randomly remove some cell types?
Good question. They tested this by randomly excluding 20% of cell types and seeing how much the results changed. The good news is most methods seemed pretty stable. Their performance didn't drastically change.
But some were more sensitive.
A few vulnerabilities did show up. For example, in vertical integration tasks, Matilda showed higher sensitivity. Its performance ranking could shift quite a bit depending on which subset of cells was included.
So maybe you need cleaner data before using Matilda.
That could be a takeaway, yes. Be more careful with data prep for methods identified as more sensitive. On the flip side, MOFA plus foo, despite being computationally heavy, was found to be exceptionally reproducible in its feature selection results, confirming its stability even with those kinds of data perturbations.
Okay, reproducibility is its strong suit. Finally, looking back at the whole benchmarking effort, did the researchers offer any big picture caveats, limitations, or key learnings about the evaluation process itself?
They did, and these are really important. First, they stressed the critical importance of using real-world data for benchmarking.
Not just simulated data.
Right. They found several methods looked great on the clean, simplified simulated datasets, but then really struggled or even failed when faced with the noise and complexity of actual biological samples from experiments.
So simulated results can be misleadingly optimistic.
Dangerously so sometimes.
Yeah.
Real-world validation is key. Second, a fascinating insight about classification.
What was that?
They found that how well you can classify cells after integration depends almost entirely on the quality of the integration itself.
Not on how fancy your classification algorithm is.
Surprisingly, no. They found that a simple standard classifier like a basic multi-layer perceptron, an MLP, performed just as well as more specialized, complex classification models in most cases.
Why would that be?
Because if the integration method did its job well, The resulting embeddings, those low-dimensional cell maps, are already highly structured, and the cell types are already well separated. The classifier just needs to draw the lines.
So focus on getting the integration right first.
That seems to be the message. Good integration makes downstream tasks easier. Hashtag #HashRotel.
Wow. Okay, that was a deep dive indeed. There's a ton of information there, but I think the main takeaway for me is pretty clear. There's no magic bullet, right? No single best method for all single-cell integration problems.
Absolutely not.
Hmm.
It's just not realistic given the diversity of the data structures and the different goals people have.
So the choice really has to be tailored.
Completely. You, as the researcher, have to go through a kind of decision tree. First, what does my data actually look like? Is it vertical, diagonal, mosaic, cross? Do I have bridge modalities? And second, maybe even more importantly, what is my primary analytical objective? Am I desperate to eliminate batch effects above all else? Or is preserving every last subtle biological difference for clustering my absolute priority? Or maybe reproducibility is paramount.
And this study, with its big comparison, gives us a framework for making that choice.
Exactly. It provides that invaluable general guide. They have summary figures, like figure 6 in the paper, that try to give recommendations based on the integration category and the task. And crucially, they factor in not just performance, but also those practical aspects, speed and memory. It's a genuine resource.
You mentioned Multigrade seemed robust for complex scenarios, like trimodal cross-integration.
Yes, its performance across different metrics in those really challenging setups makes it a very strong contender to consider first, even if it needs more computational power. Its ability to synthesize those three complex layers effectively seemed notable.
Okay, so this leaves us with something for you, our listeners, to think about. Let's say you're faced with a real data set. It's trimodal RNA, ADT, and ATAC. It was generated across 3 different experimental batches. Knowing everything we've just discussed, especially that fundamental trade-off between correcting batch effects and preserving biological detail, which integration method might you choose to try first? And what's your reasoning? Is it raw performance, speed, robustness? What stands out to you as the most critical factor for your hypothetical analysis? Something to mull over.